David Meyer. I was a postdoc at EMBL and then later a group leader. I was educated and grew up in Southern California, getting both my bachelor's and PhD from UCLA. I moved to the Biocenter in Switzerland, in Basel, for 5 years as a lecturer and researcher before I got a tip from Brigitte Jokusch, who was a group leader, a new group leader in EMBL at the time, that there was this guy, Bernhard Doberstein. And Bernhard had a horrible experience in that an MD came to him as a postdoc, within a week realized that research wasn't for him, and left. So there was an opening for a postdoc, and Brigitte said, listen, come on up to EMBL. There's so much here happening in Heidelberg. So I did, and I met Bernhard. And we hit it off immediately because I was very interested in his work. I had read about his work in the Blobel laboratory, and it was fascinating. It was a whole new molecular approach to understanding membrane biology, which is exactly what I was doing as a postdoc in Basel, but not with the level of sophistication and in vitro systems that Bernhard had worked out in Günter Blobel's lab in Rockefeller the few years before he came to EMBL. There's a thank you that if Brigitte ever watches this, someone who has a very special place in my heart and was a great colleague, but more importantly, initiated this whole chain of events that we're talking about today.
So that's a fantastic CV, David, and a great resume of the time you spent at EMBL. I think one of the things that most of us, it's certainly true for me, who worked at EMBL, feel is we were there in the golden years, but you were there even before me, and I'm sure you feel that as well. So perhaps you could paint the picture a little bit about what was EMBL like back in those days at the start of the 80s, and also what was your field like?
Well, the good news is I arrived shortly after the building on Buxberg opened. Previously, it had been a floor of the German Cancer Research Center, the Descartes, set down in Neuenheimer Feld. So they all moved, and this was brand new. There were labs that were completely unoccupied. This was, to use kind of an analogy, the Wild West, as we would say in the United States, because there was so much freedom. A lot of what had to be instituted wasn't instituted. This led to the ability to do just about anything. And EMBL was on the cutting edge. We had some people there who I'm sure you've heard of, like Yanni Nusslein and Eric Wieschaus, now Nobel laureates, Jacques Dubochet, now Nobel laureate, and so on. And these were people who were doing the pioneering work in Drosophila and in cryo-EM, respectively, that ended up getting the Nobel Prizes. So the atmosphere was absolutely stunning. The other thing was you could get anything made you wanted to. There was a workshop with qualified industrial experts who could build anything at any time, from a pen holder to a gigantic automated electrophoresis setup. It was just unlimited, and everything was possible. And that's why it was so successful. But it wouldn't have been without certain people. And Sir John Kendrew, who was the head of the institute, the director at the time, was pretty laissez-faire. A lot of the time he wasn't even in Heidelberg. And so people like Kai Simons, who was the head of the cell biology program, and the people who were in charge of structural biology, the people who were in charge of the computing, and people who were in charge of instrumentation, which was another aspect of EMBL, really were able to do the things that were necessary. And as far as cell biology is concerned, Kai put together a consortium of people, including Bernhard Doberstein, including Graham Warren, including Ari Hellenius, including Daniel Luvar, all of whom went on to be, I hate to say, like myself, institute directors and full professors at major universities and major contributors. It was just amazing. The EMBL situation from the time I got there in '79 and continuing through the time after I left in '87 was just the birthplace of a type of cell biology based on the results that were pioneered, which includes the use of viruses, which includes the use of in vitro systems, that really set the stage for major discoveries and major Nobel Prizes down the road. Günter Blobel, Randy Schekman, Jim Rothman, all of these people who went on to win Nobel Prizes did a lot of work that really came out of some of the studies that were done very early on at EMBL at the time. So for me, it was just heaven on earth. I could do what I wanted, and I was a postdoc for starters. It was the best time scientifically that I've ever had. I worked shoulder to shoulder with Bernhard. I worked with my own hands and was there 24/7, coming in on early Saturday morning to expose and to develop the gels that had incubated in the freezer overnight with film and be excited about the results. And as I said, 3 cell biology papers resulted then in the discovery of a new protein important in membrane assembly and protein transport within the cells, which turned out to be not a letter, but an article in Nature. And that really set my career off. And I have to blame, if you will, or at least credit the responsibility for my success to what I was able to do at EMBL. I would not be here today. I would not have accomplished what I accomplished either at EMBL or at UCLA without what I gained from my experience at EMBL.
There's so many things to unpack in what you just described, because there's a great model there. There's a blueprint for how to do great science. And yeah, I think it's important that, you know, in these EMBL chats that people like yourself can explain that and put the message across, because I don't think we see that still amongst the funding organizations, that it's automatically understood that that is a great way to do science. And I think we have to amplify that message. People like yourself need to be there shouting about this. I think the other thing you've touched on very inspirationally was not just the money or the resource, but the people in the esprit de corps. Because having that feeling of, you know, we're in this together, we're a team. I never experienced myself, I'm interested in your opinion, I never experienced rivalry between the different scientists, rather massive opportunity for cross-fertilization and collaboration. The biggest pressure I found was from peer pressure, from seeing how good the other people were. And that made me want to work harder.
I completely agree. And like I say, I was plugged into the cell biology group from the start. I occupied a half a bench, and Bernhard's group occupied one whole bench, which I had to split with another alum who was a postdoc of Bernhard, Sunnequist. And the other half of the lab was Ari Helenius. And this is when Ari was doing this groundbreaking work on endocytosis and how viruses got into cells, which of course, receptor-mediated endocytosis is all based on Ari Helenius's work. Brown and Goldstein, great, but you know, let's give Ari credit for what he and Kai did. And Kai was right next door, and Henrik Garoff was a postdoc in his lab. And I have to say that this was exactly as you put it. This was non-competitive, friendly, inspirational collaboration. Talking your results, whether they were good, bad, or ugly, never posed a threat, because you knew that these folks were interested in helping you, and they knew that if you were successful, they would benefit from it as well. That was the cell biology program at the time. And there were people who actually loved to give you advice. And hats off to Graham Warren, who knew everything about everything. And you could talk to Graham and he would give you ideas. And the first paper that came out from Bernhard was actually a Nature letter in collaboration with Graham Warren, using an in vitro system that they had devised to determine that there were proteins that could be isolated that were involved in the process of translocation across the ER membrane. And this was Graham, but I still remember sitting in Ari's office when Bernhard, which was shared with Bernhard, when neither Ari or Bernhard were there, sitting with Kai. and going over my results. And he would tell me, David, this is what you have to do next. David, don't ignore this. David, you have to move forward on this. And I remember I was at the time that gene cloning came in, and I'm that old, and a lot of people were getting into it. In fact, the cell biology group had hired someone, Keith Stanley, who was sort of the representative cloner. And I wanted to continue to do biochemistry. I wasn't into nucleic acids. I wasn't into molecular biology. And I was scolded because I didn't realize at the time how important it would be 5-10 years from then to actually have cloned the gene that encoded the protein that I had discovered. And of course, he was absolutely right. And he was telling me to do this, knowing what was coming down the pike and helping me get there, which I ultimately did. And that was the spirit of giving advice, giving criticism, but collegially and in a friendly manner, non-combative, and totally productive.
So maybe you can recall for us some of the specific experiments or breakthrough things that you did yourself or that you saw your colleagues do that made a lasting impression on you.
Well, I would say that aside from going into detail about the individual experiments, what was the most motivating was that Bernhard, my mentor at the time, was there next to me at the bench. And he was doing experiments. He was showing me how to do it. He was showing me how to isolate ribonuclease inhibitor from human placenta long before you could just buy it. In fact, when we started, the only kit that you could buy, and it was ridiculously expensive, was reticulocyte lysate for doing in vitro protein synthesis. And you had to buy the kit and you had to buy the isotope with it, and it was ridiculously expensive, but that was the only thing. Everything else we made from scratch. We poured our own gels. We isolated our own RNase inhibitors. My God, what a contrast from my previous mentor in Basel, who was a politician. He was rarely in the lab. As a matter of fact, he was on every Swiss commission, their science foundation. He was traveling the world. There's a famous story, and I got to tell this, and I won't mention any names, but he was such a well-known traveler. He got to the airport in Basel, and the guy behind the counter said, "So where's," in German, of course, Swiss German, "So where's it going today, Herr Professor?" And he's doing this. And he says, "I can't seem to find my ticket." He said, oh, just tell me where you're going, and I'll issue you a new one. And he says, "That's the problem. Without my ticket, I don't know where I'm going." So Bernhard was like boots on the ground, right? He lived right down the hill, and he was in the lab as much as any of us and is excited about doing the experiments and getting the results and participating in them. And that was just, for me, the inspiration to do the same when I became a faculty member, when I had postdocs, when I had graduate students. If you can do it, teach them what they have to do, work side by side with them at the bench as much as possible.
What would you say was the best advice you've gotten along the way, either from EMBL or afterwards?
Well, I would have to say that I had published, I don't even know if my cell biology papers had come out, but I was onto something in Bernhard's lab. And I thought, okay, I'm going to go out and I'm going to apply for all these assistant professor jobs. And this was like a year or two after I got to EMBL in the United States. I got 9 interviews, everything from University of Virginia to Caltech. And I went out there and gave all of these results, which weren't published and were very promising. But, you know, the beef wasn't there yet. And I was then advised, David, look, and this was from, you know, Sky and the others, you can't go out and try and sell yourself just because you think you were onto something really great. They're not necessarily going to buy it. You have to have a story. And this is what Graham Warren taught me, especially when I'm giving a talk. You should tell a story and there should only be one. How many times do you hear people say after 20 minutes into their talk, I'm now going to switch gears? And then they talk about something completely different, just to show you that they're not just doing this in their lab, but they're also doing that in their lab, or even a third thing. Graham's best advice was, tell a story and only tell one story, because if you get to another, they're not going to remember the first. And this way you can hold their attention span longer. And the other thing he taught me is how to write. Subject, verb, object. Don't become Germanic in the way you write. When you talk and when you write, short sentences, very well defined and specific. So I have to say, in terms of speaking and in terms of writing, the best advice I got was from Graham on how to do that. And I am absolutely convinced that my ability to do that, and I passed this on to my students and postdocs, was largely responsible for my invitations to international meetings, the success of my publications, the need not to do extensive revisions, except maybe an experiment or two in my publications. That would be it.
What advice would you like to pass on to young scientists today?
Yes. The first thing that I would say is that you have to be rigorous. Too much of what goes on is just trying to hit the home run. I see this in young scientists all the time. And the more they try and the more they fail, the more they try to hit the home run. 99% of all the experiments that you do only lead to knowing what the next experiment is that you have to do. And unless you do that methodically with the right controls, you are going to fail. And it's going to put you in a place where you're going to become more frustrated and more desperate. So from the very beginning, plan things out one step at a time. You want to go from A to B, not from A to Z. And in that way, you have a much greater chance of succeeding. You'll have a notebook that has everything in it that you need. You'll be able to go back pages and see exactly what you did then and why it's not working now. And that's really it. I tell this to postdocs who think, oh, they've got to get a Nature paper, they're not going to get a job. I tell this to graduate students who think they have to get a Nature paper, they're not going to get a good postdoc. They have to try not to win it all with one swing.
Do you think the system contributes to that feeling that you have to hit a home run because it's very intolerant of what's perceived as failure? They want short success.
I think people see that people have hit the home run, but they don't understand how they did it.
Yes.
It's what we call in baseball here small ball. And if you understand baseball, that's, hits, singles, doubles, walks, and so on. In the end, you score runs. So that's the way it has to be. It has to be small ball. Even though you might see, like I had a graduate student, an MD PhD student in my lab, published an article in Nature. But it was painstakingly slow step by step, and that's it. Many people might think, oh, look, this guy, MD PhD, hit the home run ball. No, he didn't. It was small ball.
Absolutely. So, you had the experience of working in the very early days of the EMBL, as it was an embryonic institute developing, went through puberty and adolescence and has grown up and blossomed. And we're now celebrating this year, as you know, the 50th anniversary. Could you maybe just sum up in a few words what EMBL means to you?
Sure. EMBL means success to me. Without EMBL, I can say with 100% certainty, my career would not have been what it became. The success that I had early on after leaving EMBL, all the way up to the job I just retired from, was inspired by the people, the institute, the collaborations that I had. That's what EMBL means to me. Irreplaceable is the one word that I would use to describe it.
EMBL and the spirit of EMBL, my opinion, is all about inspiring and training new generations of scientists. And then a really fundamental part of that mission is to allow them then to go and take that inspiration and training back to either their home country or to go on to new laboratories and to pass it on. What would you say, David, would be your proudest achievements in your career post-EMBL?
Long about 1983, 84, John Tooze decided EMBL should have a PhD program. And I was a big advisor for that. Why? Because I had an American PhD. No one else did at the time. And that was the model that he wanted. We admitted the first, I believe, 6 students, something like that, all of whom have gone on to great stuff. And full professorship at the University of Michigan and others. This was our crop of, first crop of graduate students. And John then anointed me Dean of Graduate Studies. So I was group leader, Dean of Graduate Studies, EMBL Heidelberg. Why is that so important? Because when I got to UCLA, I was in the medical school and we had 6 different basic science departments. Each had its own PhD program. We have the College of Letters and Sciences with 6 more departments, all of which had their own PhD program, and they were all doing cell and molecular biology, all life sciences. So what I did at UCLA was bring them all together, and that was politically very difficult, particularly between the medical school and the college, where there was a lot of animosity because the physicians were getting paid more than the scientists in the college, and the usual story. And I brought them all together. It took me a year and a half to create a program called UCLA Access to Programs in the Molecular and Cellular Life Sciences, which existed for 20 years and brought together 12 different departments for common recruitment, common admission, common first-year curriculum. And this became a model for interdepartmental programs across the United States. And I'm very proud of that. And that was inspired by group leader, Dean of Graduate Studies at EMBL. And I would say that is one of the major accomplishments that I pulled off and I'm most proud of.